Hello, I'm Rahul Singh. I'm a PhD student in biomedical engineering. My research is about developing materials to help regenerate tissues in the body. In particular, I'm focused on regenerating very small blood vessels. So, let's take a quick overview of what I'm talking about. This is the cardiovascular system. It is composed of your heart as well as blood vessels which carry blood away from your heart called arteries and blood vessels that return it called veins. Where these two meet are capillaries. Capillaries are the very, very small blood vessels that allow nutrients and oxygen to leave your bloodstream and nourish various tissues. So, why am I concerned with studying ways to come up to repair capillaries? Well, that's because capillaries are involved in a number of vascular diseases. One example is stroke, which results from a lack of blood flow to regions of the brain. Another example are heart attacks, which result in a lack of blood flow to various regions of the heart, and diabetes, which can lead to peripheral artery disease and a lack of blood flow to your hands and more often your feet. So this lack of blood flow prevents the tissue in your body from being nourished, and it eventually dies and leaves an ischemic region. So what I'm focused on are developing new materials and cell-based therapies to re-engineer this tissue. So there are a number of approaches towards creating an engineered tissue. In particular, I'm going to go over one which combines materials and cells into a new structure which can be reimplanted into the body. So, we start with taking a very small sample of tissue that we're trying to recreate in a process called cell isolation. We next expand this very small number of cells into a very large number of cells that we can use to build a large tissue. We combine these cells and materials in a very specific way to create an engineered tissue, and then we allow that tissue to mature outside of the body until it's of the appropriate size and developmental stage that we can use it in our therapeutic application. So, my research focuses on a small aspect of this process. I'm focused on creating an engineered tissue, and in particular, I'm focused on materials that I can use to create the tissue that I want, which are capillaries. So, picking a material depends on what tissue we want to recreate. Here we can see a number of various cells that we could find in these different tissues. In the brain, for instance, we find neural cells and endothelial cells, whereas in the heart we find muscular cells, and in a foot we'd find bone and cartilage. So an important thing to keep in mind is that all of these cells want to grow in an environment that mimics what they were originally growing in the human body. So what you can see now are all these various cell types mapped out according to the type of material that they want to grow in this case plotted with respect to how stiff those materials are. And we want to pick one that matches the tissue that we're trying to recreate. So for an endothelial cell, we want to pick something that is very soft. And in terms of engineered materials, we've got options between hydrogels, plastics, and metals. So hydrogels are very, very soft materials, so we want to pick one that matches what these endothelial cells grow in. So this is something that is sort of a key concept in tissue engineering, is we try to recreate the environment that these cells originally were in order to allow them to reconstruct the tissue that they were originally taken from outside of the body. All right, so we want to use a hydrogel to help recreate an environment for endothelial cells. So it's a good question to ask would be, what is a hydrogel? Normally we don't encounter these materials. The closest example that uh, we normally see of hydrogels would be things like Jello. So they are, in fact, materials that are 99% water and a very small component of something binding all that water together. So here is a picture of one of my constructs. So this is a hydrogel. It's about a millimeter across, and as you can see, it's transparent and it looks like a little blob of goo. That's because it essentially is a little blob of goo. But if we look inside the hydrogel, we can use a high-powered microscope as well as stains to highlight the capillaries that have formed inside this material. And in this we see in red a capillary produced of multiple endothelial cells that has the appropriate structure that we were looking for. So while we were able to create a capillary in this material, that's still not an engineered tissue. To go one level further and scale this up into something that would eventually produce a therapy. So in this case, we go back to our original diagram, and we've already decided that the material we want to use are hydrogels. Now let's look at making an engineered tissue out of it. All right, going back to our description of tissues, we know that they are diverse. They're not just composed of one structure. So I've been talking on about capillaries, but they're not the only component of a tissue. They're just the component that deliver blood. So we've come up with something that has endothelial cells and capillaries. But say if we were to create a therapy for stroke, we'd need something that has the endothelial cells and also neural tissue. Or if we were to come up with a therapy for our heart attack, we'd need something that has endothelial cells and skeletal muscle, or cardiac muscle. And if we were to recreate a therapy for peripheral artery disease, we'd need our endothelial cells as well as muscle or bone. So how do we go about recreating primitive structures like this outside of the body? The approach that I use to recreate these tissues is a printing technique in which we build up the tissue by adding different materials together as well as our cells. So starting with a base, so in this case a slide, we'd add a liquefied form of our hydrogel. I then use a mask which has areas that are blocked out as well as areas that are transparent, and I put that on top of my liquid. I then shine light on it. This selectively solidifies different regions of the material, and then what I'm left with when I wash out the unsolidified material are just little patterns in the shape of my mask. So the next stage is just adding in more hydrogel, this time with different properties, as well as the cells that will eventually become capillaries. And then again cross-linking, or again exposing it to light, and then adding in some supportive cell types. So then I put the whole structure that I've created inside a dish and let it mature for some period of time. So through this process, I'm able to pattern the original material that allowed capillaries to form, as well as another material which closely matches the tissue that we're creating a therapy for. So after these tissues are allowed to mature for some period of time, I get something like this. So what these are an array of small regions of capillaries as well as the supportive material that I've put around it. So these are a mimic of what you'd find in the body, which would be capillary beds. So if we zoom in on them, we can see that they're indeed composed of the same endothelial cells in a little tube-like structure as they were in the other material. So going back to the whole process, we've now seen that we can combine our hydrogel and cells and get a capillary. We can structure this into an engineered tissue, and then we allow it to mature into little beds. So the next stage would, of course, be a therapeutic application. So my research doesn't focus on translating this into the clinic. There are other people who are specialists in terms of turning these early stage devices into an actual therapeutic application. So that's an area of ongoing work for other groups. But ideally, what I hope is that through this work, we're able to create vascular therapies wherein we could take these dead regions of tissue and simply place a little vascularized patch on top inside the tissue and allow it to help regenerate what was originally there. And I would like to thank